Well, to use the simple starting metaphor, the world of knowing and not knowing and crossing this chasm to doing, it really left me with a sort of an better informed chill, but not a great deal of or a sense of how we get across that great chasm. Dr. Omenn is going to speak about molecular diagnosis and take us deeper and further along the journey of healthcare delivery.
Okay, well, thank you, Scott. Larry, thank you for inviting Martha and me to the meeting. It's been fascinating. And I'm thrilled to be paired with Frank Prendergast, a real important leader in this country in cancer research and a leader at Mayo for so many decades, and a personal friend. We're great to see you here. Okay, I'm going to talk about molecular approaches to diagnosis and prognosis for patients with cancers. We always have to remember we are treating patients, not just cancers, and that, as we heard beautifully described, we should always be talking about cancers in the plural. It is a part of the problem in our communication about progress about cancer research and clinical applications, that we give the impression that there might be a great magic bullet, or that one particular approach is going to solve all these many different diseases. Cancers are different in their origins, in their progression, in the response to therapies, and in their effects in different patients. We're supposed to disclose, I guess at university, probably here too, involvement in the biotech industry and various other companies. It's actually a very big part of the aim to bring academic and other research findings to practical fruition. These covers you've seen from other speakers. I hope you have them because they're collector pieces. And they were presented on the same weekend, almost exactly 10 years ago, 10 years and 3 months now. The public and private sector competing efforts to sequence the human genome. And here are the major players. Everybody's heard of Craig Venter and Francis Collins. Some of the other characters not as well known. The guy whose technologies were absolutely essential to the human genome is shown in the lower right is Lee Hood. He and his team produced the DNA synthesizer and sequencer, the protein synthesizer and sequencer, miniaturized, made it feasible to use very small samples, and very reliably perform these molecular chemistry manipulations and identifications. Eric Lander at the top is one of the several big genome center directors in the United States. There are now other such players around the world, as we've heard, and he is also a member of President Obama's Presidential Council on Science Advisors. Least known, perhaps, is Ari Patrinos, and perhaps little remembered is the role of the Department of Energy, which stepped up to grab the reins about pursuing the human genome at a time when NIH was really not interested, and where the prevailing wise people in the field said this is just routine drudgery. Besides, it will be infeasible. It'll knock away funding for everything else that we should be doing. And it turns out we were able to do the genome and all the other research and greatly amplify what we can do with technology to ask better and newer questions. In fact, it's one of the basic principles of the history of science and technology that the old linear model, from basic research to applied research to development to applications, is inappropriate. DeSolo Price, a prominent historian at Yale, among others, pointed out that technologies are often absolutely essential, not to just changing what we can do, but even what kinds of experiments and insights we can conceive. So this is the history in genomics. Now, there was a tremendous media coverage in the year 2000 when Prime Minister Blair and President Clinton announced the progress on the genome, and the next year when these two issues of the sequence and many applications of the sequence were published. 5 days later, we've heard a lot about proteomics and other omics at this meeting. 5 days later, the Financial Times of London ran this wonderful cartoon. Where is DNA? Off stage, off in the shadows, Center stage, protein. And as we just heard this very last few minutes, the action molecules, most of the action molecules of the cells are the proteins. These are enzymes, receptors, structural components of cells, hemoglobin, collagen, and of course antibodies. And at that time, people were impatient already that we should have proteins, proteomics methods to capture all the proteins, just like presumably all the genes had been pretty much captured, sequenced, and put it to good use. Now, impatience is an American trait. Here's an example. In 1965, then President Lyndon Johnson went by helicopter from Washington, D.C., all the way up the street to NIH, and swooped into the site, applauded the prowess of biomedical science and scientists, and reminded us that the nation is waiting for results from research. Not just papers and grants and trainees, but better medicines and better health. And that is truly our mission. What can we do to accelerate the impact and the benefits of research? Well, we've heard a lot at this meeting. One is to be bold about our aims and our technologies. Second is to create resources and organizations, as we heard about Genetic Alliance and others that really do drive and assist the scientific effort. To feel a passion for the mission, to really want to make a difference and not just have a work-a-day job, and to measure, conduct research on what the impacts are for the populations, and importantly, for the economy that depends on new science and technology. So a lot of that new science is captured in this figure here, which is the same theme we've heard from others. You can start from the genome and work toward the clinical features, traits, diseases of individuals and populations, or you can start from the problems, the genetic and health, the health problems, clinical problems, the phenome or phenotypes. Think about the chemical and physical and biological exposures from the environment, from the diet, and work our way down toward proteins and genes. Try to understand the clinical disorders, the traits, and what we can do about them. We have a construct Frank just mentioned about computational approaches and computational medicine. We start with data. We're generating avalanches of data. But our goal is to translate data into real information in organized, usable forms, shown here from the bottom up, genomes, transcriptomes, that means the messenger RNAs that take the message from the genes to synthesize, guide the synthesis of proteins. And then proteins work in pathways which quite often end in metabolic pathways like glucose metabolism, or amino acid metabolism, nucleic acid metabolism. And we can capture that in a knowledge base which is organized around concepts, modules, networks, where we should be able to eventually understand what's going on at a cellular and organismal level. This is a slide from colleagues at Vanderbilt that, for the longest time, doctors would say, only with these hands, Mrs. Jones, did you have a chance of surviving your cancer. Or doctors who said, in my experience, this drug will or won't work. Now it's supposed to be guided by evidence. It wasn't many years ago, I gave a talk to the big Rotary Club of Seattle and talked about evidence-based medicine. And at the end of the talk, somebody asked, doctor, if this is new, what have you guys been doing up until now? Well, now we're trying to make sense out of all our molecular understanding, out of trying to link together these many levels of inquiry and observation and measurement, and that's what we're going to have to learn how to explain in forms that both doctors and nurses and patients and their families can understand to make life-influencing decisions. So here's a summary of what I'm going to talk about. First is that molecular analyses of cancers already can reveal information about mechanisms of initiation and progression and provide a foundation for clinical tests. Tests have to be organized around clinical utility, the scenario of use. So the use can be making the proper diagnosis when there's a differential diagnosis and it's uncertain. Earlier diagnosis, and for certain cancers we know that earlier diagnosis is critical, like ovarian, pancreatic, lung cancer, because we really have very little to offer late-stage diagnosed patients. Third is the prognosis and the risk of metastases. For other kinds of cancers, actually the prognosis is much more important than an early diagnosis. In fact, all an early diagnosis achieves, for most or maybe even all of those additional patients, is to carry the stigma and diagnosis of cancer a lot more years, and to, if the metric is 5-year survival as a cure, be quite certain that you can improve the results, because you made it earlier. And this whole question about the response to therapies and having much more specific therapies to offer patients. We just heard that cancers are very heterogeneous in all aspects, and all these molecular approaches are complementary and should be complementary in guiding the construction of appropriate tests. It is a long path from discovery to confirmation, which we often skip, publish immediately, to validation by another laboratory or another group, appropriate clinical trials for tests as well as for drugs and vaccines, and then FDA approval to establish the validity and utility. So let me start with prostate cancer. The standard test for diagnosis, both in screening and for recurrence or reappearance, clinical reappearance of the cancer, is the prostate-specific antigen. It is a normal protein in the prostate. It's present in normal prostate tissue. It goes up with diseases, both cancer and non-cancer. Actually, this test works fine for seeing if a cancer has been successfully removed or reduced in mass, and to see if it reappears in the same patient. It is a terrible test, a very expensive in the aggregate, very expensive cost to our society for poor sensitivity, poor specificity, and trivial predictive value. But it is the standard of practice. It's very hard to displace, and newer and better tests under development are having a hard time displacing clinical practice. A comprehensive approach to prostate cancers, and then all cancers, is in the form of this database called Omenn, developed by Roel Chanayan and a student of ours at Michigan, Dan Rhodes. It started with all the then known microarray hybridization datasets of messenger RNAs, the transcriptome, gene expression information database, and then expanded from prostate to many other cancers, and both published and not yet published work that people would submit. And it was organized in a database that contained statistical and bioinformatics methods embedded so that you could really use this database online for discovering features of cancers of different kinds, what's known about response to therapies, and other kinds of data mining for correlations. One of the most remarkable developments from this database is shown here. This is a measure of outliers. And one thing we can celebrate in the work of geneticists for decades is to realize among all the heterogeneity that there are people at the extremes of any kind of a measured result. And quite often they are very informative. In fact, the middle ground is less informative most of the time. So what was done here was to look for extreme up or down expression of these many, many mRNAs, gene products, that were embedded in the database. And they found that a particular family of genes, called ETS family genes, was overexpressed. This is for a gene called ERG. One of that family. They're transcription factors. They drive proliferation of cells, cancer cells. And what happened is, shown in the bottom middle, which I'll now expand for you, is that there was a fusion of two genes. The 5' gene specified a sort of typical membrane serine protease, an enzyme, which is under androgen control, the male hormone which drives prostate cancer growth. The proliferation factor is normally not under androgen control at all. But when it's fused, and much of this gene is removed, and a little bit of this one's removed, makes a fusion gene. Fusion gene has the properties of the transcription factor, and the control, the regulation of the androgen driving the promoter of the first gene. And this turns out to be 50, 60, maybe 70% of prostate cancers. Totally unanticipated result, but it came out of mining these data. Now, fusion genes are not unknown. They are, in fact, at the heart of the chronic myelogenous leukemia, chromosome abnormality, and the Gleevec treatment that was mentioned earlier. In fact, they're common in leukemias and lymphomas and a few other cancers. Not cancers, sarcomas. But in carcinomas, epithelial cancers, they were essentially unknown or extremely heterogeneous and miscellaneous, thought to be of no consequence. That is not the case for the prostate. Now, a tremendous amount of work has followed from this original publication in Science in 2005, and we now know that there are combinations of the first gene and the second from these families, and they have this effect of driving proliferation of cells, making prostate cancers, and targeting either the fusion gene and its product or something downstream in the mechanism of action of that gene complex is a viable therapeutic approach. It's also been turned into a diagnostic test. Well, then the question was, what other fusions are there? So there's been further searching with very clever methods, including the new high-throughput genome sequencing, for other fusions in prostate cancers and for search for fusions in additional kinds of cancers. I can't go into all that today, but I want to stress this point, that there are specific therapies, already approved drugs in some cases, for particular mechanisms. And Frank said we should be treating the mechanism, not the presumed tissue of origin of the tumor. Tissue of origin is sort of like a geography of cancers in the body. We should be treating the mechanism where if we have the information, the proof, the will, the nerve, treat it before it's obvious and is out of control and has accumulated many other changes which make it much harder to treat. So this approach has been taken, and there's a gene called SPINK1, which turns out to be in 10 to 15 more % of patients with prostate cancer. And just in the last 2 years, young people working with Chinayan and with me have produced fusion gene evidence for 1 to 2%, a very small percent, but there are 200,000 cases per year in the United States. So that's a lot of thousands of patients, for which there are already known approved drugs for those targets. So this suggests a totally different approach. Instead of discovering what's awry in a particular cancer and trying to see if you can figure out how to attack it and whether you could create a drug with all the 15 years of drug development to follow, you take a drug that's already available, has known properties, maybe not everything is known about it, but some properties are well established, it's FDA approved, and you can see if you have found patients for whom the drug will be specifically useful. Find the patients to match the drug. This is going to be a very productive approach, and these are real examples. Now, if you take a patient-centric approach for prostate cancer, what you really, really want to know is, since a small percentage, 10%, maybe 15%, account for almost all the mortality, those which are invasive and metastasize, what are the molecular markers and mediators for such cellular behavior, metastasis, invasion, movement? And how can we tell apart the lethal cancers from the relatively innocuous cancers that look the same in the microscope and by staging clinically? This is actually a lot more important, I feel, than earlier detection of that large number of cases. Here's the story that follows. We combined the approach of the transcripts and the informatics analysis of what pathways and networks were involved with the transcripts. We knew a lot about antigen-driven pathways, but we're looking for other kinds of things. And metabolomics. This is another kind of mass spectrometry method. We'll talk about mass spectrometry for proteins in a minute, but this is looking at small molecules, amino acids, nucleic acid, sugars, lipids. And what you can see here is that in the benign prostate, there's very modest variation around the vertical. In localized prostate cancer, there are quite a few outliers here. A little hard to see maybe with the bright lights, but you can see it easily here in the metastatic. There's a tremendous amount of variation of some of these several dozens of compounds that can be reliably measured and identified. One of these, among half a dozen that we studied, was particularly striking, and it was focused on because the concept maps that we had lit up amino acid metabolism and methylation metabolism, two kinds of pathways, networks. We said, well, what could possibly be a connection between these two? Neither was expected from previous information. Well, there are chemicals that are methylated amino acids. And the simplest is methylglycine. Glycine is the simplest amino acid. One methyl group makes methylglycine. It has a name. It's called sarcosine. In the normal prostate, we could hardly measure it. In fact, we had to develop a more sensitive assay to be sure we could even measure it. In the localized prostate cancers, it was readily measured in most of the cases, but at a very low level. In the metastatic prostate cancers, it was sky high or very much increased in practically all the cases, as shown here. Now, so far, this is just what we call a biomarker. It's an association with the state of metastasis. So what Chennaiyan's lab did was to take cell lines, prostate cancer cell lines, that were very invasive and not invasive at all. The not invasive at all were fed sarcosine in the medium. Sarcosine level went up in the cells. They started moving, invading. You put them in a mouse, they made metastases. You could give the precursor, glycine. It would be methylated, same thing. You could transfect the gene for producing the sarcosine, the N-methyltransferase. Sarcosine goes up, cells metastasize, spread metastasize. Or you could block the degradation of the sarcosine dehydrogenase. So every step was done. Every step had the predicted result, much more invasive, metastatic in animals tested. You could start from the sarcosine in the invasive cells and knock it down, which was done, and it became non-invasive and non-metastatic. So we believe we have found not just a marker, but a mediator. And this question was asked by Greg or somebody yesterday about other work. This is a real mediator and looks very promising, both as a diagnostic and as a target for managing most aggressive prostate cancers. It's published in Nature just 2 years ago, and lots of work has come from this. So I think that's a pretty exciting example. Next, breast cancer. Same kind of approach, gene expression signatures to guide, in this case, treatment decisions. And the big question that many, many women need to address is whether or not to use systemic chemotherapy after initial surgical treatment for breast cancer. There are a lot of side effects, as you all know. It's been mentioned by many speakers here. And the question is it worth the risk to try to reduce the last little bit of recurrence and possible mortality from the tumor that was thought to have been removed? So this work was led by a woman in Netherlands, Laura van Veer, and her colleagues. It published in 2002, specifically on node negative, that means no spread, stage 1 or 2, very early stage breast cancer, women under 55, trying to distinguish those who would develop no metastases over the next 5 years from those who would, or in fact had, with retrospective studies. And they started, well, this is what you do. You take the surgical tissue in this case. This is not a blood test. And you do these assays, just like I showed before for prostate, massive microarray hybridization, thousands of probes, in this case 25,000 probes for genes and their expression as mRNA from the tumors. And from that, they chose the 70 that seemed to be most informative. So it's a balancing act, picking a panel of multiple markers, because you want to have enough to find the variety of cells and tumors reflecting the heterogeneity. At the same time, you don't want to have so many markers that you pick up a small percentage of, for each one of them, of normal, or in this case, won't progress, and mislabel them falsely positive as likely to have metastases. Well, here's the result. This is an unusually good result. So at the top are women who had a good profile, and look at that survival. This is overall survival, basically 100% for 5 years. And the women who had a poor profile on the test did not do well at all. That's a remarkable separation of the two curves. If you asked a different question, which was how many had no metastases over 5 years, they survived so far, but they had metastases. Well, this wasn't quite as clean. And now you have a problem. Will the woman be satisfied to have a profile that suggests excellent survival, or does she want to make sure to drop that risk of some risk of metastasis later as far as possible? I think most women choose the second. So now new studies are undertaken, trying to say, what about if we focus on the intermediate cases instead of the ones at the extremes? Now you want to know about the confusion in the center instead of identifying the information from the clearer cases at the extremes. And this is a big, big trial in Europe using the 70 gene signatures. So in the center here, these are the discordant cases. High risk based upon the clinical and pathological findings, low risk from the gene, versus low risk on clinical and pathology versus high profile. And you have a choice to use one or the other. So these data are being collected currently, and the expectation is that so far the 70 gene profile seems to be the better guide. But they're expanding the study, doing additional studies. In general, we never have a study really big enough to answer all the possible questions with extreme statistical confidence. This is a study with 6,000 women. Quite a big investment, important question. Alongside that one, there's another test. Both of these are FDA approved. This is called Oncotype DX. This is a 21 gene panel. This is not just the highest ranking genes, miscellaneous genes. This was chosen by sets of genes that would reflect different kinds of breast cancers. Estrogen receptor positive, estrogen receptor, another receptor negative, HER2 new amplification positive or not. And it generates a risk score, not just high or low like the previous test. And the answer here is that you either get a low score or a high score, which suggests high risk, or intermediate. Now you're back to what do you do if you're in the intermediate category, same kinds of questions addressed in that previous study for the other test. This is launched by Genomic Health. The previous was by a European company, now with a California address also, called, well, it doesn't matter, Arcacia. Same approach can be taken to every other kind of cancers. So here's another example. Let's have a couple slides about lung adenocarcinomas. And this is a particularly interesting study because it was the bringing together of findings from 4 different populations, 442 patients total, using 8 newly developed prognostic models for what would be the survival picture going forward for men and women with early stage lung cancers. And the result is shown in this slide. The curve you're interested in is the high score predicting need to intervene aggressively because of high risk. And that means lower survival. So the light blue curve should be the lowest result. At the top, it looks great, right? The risk is that somebody would publish only that. And there is a practice in the world of giving your best results. These other 3, that looks pretty good, not nearly as clear. These 2 down at the bottom show no difference. So here we have classifiers of different performance with different data sets, and still, I would say, considerable ambiguity about how useful they will be. Let me turn now to proteomics. And the overview of this is that we want to study proteins. We can profile tumor specimens, as I just showed you we did with transcripts and metabolomics. We can profile blood samples. And from the blood samples, we can look for prediction or response to therapy, prediction of recurrence, response to therapy. Here's the first FDA-approved example. It's called the OVA1 test for ovarian cancer. Any of you heard of this? It's a not very satisfactory test. It's almost embarrassing. But it's a great example of focusing on a very narrow question, narrow indication for clinical decision-making and getting FDA approval. Because the indication here was for women who are about to undergo surgery for a known pelvic mass to guide the surgeon, typically a general surgeon, whether it's likely to be benign or cancer. And statistically, 80 to 90% of these would be benign. So this is a useful bit of information. I checked this out with a wonderful gynecologic surgeon named Beth Carlin at UCLA. She says she's never used the test, that she's a gynecological surgeon. She's always prepared. In fact, she would almost always get the cases that are most likely to be cancers, and if it turns out to be benign, that's a blessing. Okay, this is the reason I say it's awkward is that it's 5 very abundant, easily measured plasma proteins that have nothing whatever to do with specifically ovarian cancer. They have something to do with chronic inflammation. And inflammation is a feature of many cancers. We're coming to appreciate it much more as a feature of cancer. Maybe it is more relevant than originally thought, but it is not specific. Nevertheless, it was developed carefully at Johns Hopkins. The tests are reliable, and they do what they set out to do. Next is work from Rachel Ostroff, who is here. I believe that's you, yes. I'm hoping to sit down with you later. And Larry's whole group, Larry from the SomaLogic. And this is quite dramatic. This is instead of using mass spectrometry to identify peptides and infer the proteins, or using antibody arrays to detect the tumor antigens, using this distinctive method of aptamers and identifying proteins through these particular clever molecules. And you can see here they started with 1,100 proteins that they could detect. They boiled it all the way down to a panel of 12, again trying to optimize between high sensitivity and high specificity. You can never get 100% of both of those parameters for real. And if you look at the descriptions here, there's things about cytokines and about angiogenesis and about cell adhesion, obviously important functions for cancers and for cancer cell growth. The statistics look remarkably good. This is a complicated thing to interpret. It's a training set in blue repeated with a test set of separate samples, and an area under the curve of 0.9 approximately is exceptional. If you run PSA in such a test for prostate cancer, you would get an area under the curve first of about 0.6. That includes those who have 0 and very, very sky-high values. If you take the range that really matters, it would be right on the diagonal, which is the flip of the coin. There are details here about different subpopulations of this study, and with different areas on the curve a little bit, and how they're put together. It's a complicated paper, but I commend it to you. It's in PLOS One this past December. Now, in my remaining time, I want to tell you something exciting about new things in proteomics in our lab. And it's particularly interesting, I think, for this reason. The Human Genome Sequencing Project began with an expectation that there would be 50,000, maybe 100,000 or more protein-coding genes in the human genome. And it was sort of a vanishing discovery of genes. We are now down to, and apparently just under, 20,000 protein-coding genes. It's considerably less than 2% of the whole genome. There's a lot of other things going on besides coding for proteins, obviously. But if we want to start with biomarker discovery, either from tumor tissues or from plasma, we have these strategies. One I've already shown you, which is to start, number one, with the microarrays, find those genes that are highly overexpressed or underexpressed in important pathways, and look for the proteins. Predict what the protein should be. The second approach is what we call targeted proteomics. Instead of measuring as many proteins as possible, use certain labeling and sequence information about how the peptides perform in the mass spectrometer, and measure just exactly those peptides as markers for those proteins that you already have evidence are informative and then can be tested on a high throughput basis and quantitated. The third is to use nature in the form of the immune response, autoantibodies, as a way to detect tumor antigens, because there are much higher levels of the antibodies than some of these very low concentration antigens. And the final is this notion of alternative splice variants. And the reason I'm so interested about this now is that we've been finding some interesting things, and splice variants contribute mightily to this business of how we get along with so few protein-coding genes. Because from individual genes, there is alternative splicing. I'll show you a picture of this in a minute, such that you get different transcripts, mRNAs, and from those translated into different proteins, which gives more action molecules from the same gene. So if we start with 20,000 genes, we may have at least 100,000 different protein isoforms before all the further variation from post-translational modifications by putting sugar groups and phosphate groups and other very important chemistry on the side chain of the proteins. So here's a picture of alternative splicing events. They are numerous. The easiest to understand is this one here with mutually exclusive exons. So either the blue one or the green one, but never both, actually appear in the protein. You know, there are exons which actually go into the protein sequence and introns, the intervening sequences in the gene and in the messenger. They're carved out to make the sequence for the protein. But you can have alternative start sites at the 5' end. You can have intron retention, which is very interesting, and alternative other options. So the short version of this is we created special databases of all potential translation products in 3 frames that could arise from the genomic information in the mouse, where we do model studies, and in humans. There are 10 million of these sequences in the mouse and 14 million in the human. And some of them are known, and more are what we call novel, that we and others now find. And that's a special kind of a database that enables this kind of analysis. And then we take the data from mass spectrometry and look for those peptides that have these special sequences in the peptide that actually include the splice junction. So we can identify that it has to be a splice variant. We can then prove it by doing a qRT-PCR analysis on the mRNA to find the RNA that specifies exactly that spliced sequence. And then we can ask questions, what could these proteins actually be doing in this particular kind of cancer? And the 2 kinds we've published recently are pancreatic cancer, where there's a model with exactly the molecular lesions that are found in 99% of human pancreatic ductal carcinomas, and the same kind of approach for HER2-neu amplified breast cancer, 15% or so of human breast cancers. So in the case of pancreatic cancer, we have found 328 splice variants that were known previously in the other general databases, 92 new ones. Example, here's one that involves an interferon receptor, and interferon receptors are important in response to certain therapies for these cancer patients. Also, the variant, the splice variant, lost cyclic AMP and cyclic GMP-dependent protein kinase phosphorylation sites, which suggest important functions that could be tested in great detail. Another example is lipocalin. This we measured. It's clearly upregulated in the tumor sample, or in the animals with the tumors, and it turns out to be overexpressed also in human pancreatic adenocarcinoma in serum, just as in the mouse model. And a couple other examples. I know our time is late, but the important confirmation is shown here, the qRT-PCR. We picked several of these and proved in every case that we could detect the message that corresponds. So the summary is we identified a large number of splice variant proteins. Given the labeling method that was used, which puts acrylamide, deuterated acrylamide on cysteine residues, only those peptides that contain cysteine are informative. But among those, we found differential expression in many. And as I said, the further work on this from Sam Hanash's lab showed that some of these were increased in human pancreatic cancer patients. HER2-neu breast cancer model, the primary data came from Mandy Palovich at Fred Hutch. It is in our Tranche repository, available for open use, and with her encouragement, we did these studies on that data set from her group. It was very interesting. They tried to find markers using antibodies, and nearly all the antibodies failed. This is a very sad story, very, very common story in this biology field. But we were able to show that there were 68 novel splice variants, over 500 known splice variants. All of the ones that we subjected to qRT-PCR we confirmed. And for 2 examples, here's a peptide that aligns to the intronic region. So this is a read-through of the intron of a gene called ROGD. It has a phosphopeptide motif which interacts with the C-terminal domain of a gene you've all heard of, which is breast cancer 1 gene. The next example is a totally different kind of protein, a transcription factor, SOX7, which also annotates to interact with the C-terminal portion of BRCA1. Now we're joining up, as I'll tell you in a minute, with our structural predictions people to see if we can model those interactions. Here's a list of several of these findings, and the purpose of this is to show that even this list illustrates multiple different mechanisms, and it's possible that different mechanisms are also clues, not just the specific splice variant and its differential expression in the cancer, but the kind of mechanism we found. There's another one we found, splice variant, differential expression, polypyrimidine tract binding protein. This actually works on the pre-mRNA splicing mechanism itself, which could have multiple effects related to cancer growth. This is using cytoscape to visualize the interactions between proteins, including, in the case of the ones, it's a little hard to see, of course, but the ones that are bolded, all of these had splice variants in our study. The ones that are bolded are ones we particularly identified as having relevant properties for breast cancer mechanisms, and they are highly interconnected here in these pathway analyses. So in this work, we have extended these findings by asking this question. Maybe I should start this way. My protein chemistry friends, and that's my background besides all the medicine and genetics, say you should study every protein in depth. This is a traditional model. It's one protein, one investigator, one lifetime. And proteomics, we generate a list of 1,000 or 2,000 proteins. And I say, well, what are you going to do with such a list? How can you even infer what the functions are? So I walked across the corridor in our Center for Computational Medicine and Bioinformatics at Michigan to a spectacular protein prediction guy, Yang Zhang, who has won the CASP competition globally the last 3 cycles, and said, have you ever done this? Anybody you know doing this? Would you be willing to try? I've picked out a couple of pairs of splice variant isoforms that I'd like you to see what your methods do for us. He says, well, sure, I got a lot of other things going on, of course. I said, well, whenever you can get to it. He said, how about if I give you preliminary results on the first pair in 48 hours? I said, I could wait 48 hours. And in 48 hours, he had pyruvate kinase type M2 for us, which has been found in the Lewis Cantley lab and now other labs around the world to be the critical enzyme in the switch to the Warburg effect of aerobic glycolysis in many kinds of cancers. And when you look at this, biochemically it was demonstrated that there's phosphorylation of the tyrosine 105 and there's differential binding of ATP. When we looked at the structures, here's green, exon 9, yellow, exon 10. Here's the overlay of the two, precisely the same. We couldn't find any difference between the structural pictures of these, but the FBP, that's the fructate bisphosphate binding, is differential between these sites, and the ATP binding, even more important, is shifted in the comparison of these structures. So we have a picture that would have predicted had we not known. It would have been confirmed now that we do know the structures from other folks. And now it turns out in the whole protein database with 70,000 protein structures, whole proteins, partial proteins, there are only a handful, maybe 7 cases of 2 isoforms by splicing that have got both forms. And we've used those as a benchmark to show how our methods work. And we've taken a whole bunch of other ones now and predicted what they should be to stimulate the people to actually do the crystal structures and the biochemistry. And we're open to collaboration on these. We'd love to see this kind of work carried forward. So I'd want to thank my colleagues and our group at Michigan at the top, pancreatic dataset, breast cancer dataset, and the prostate cancer work at Michigan also. It's a pleasure to be here. Thank you very much.